This method can answer key questions in the transcriptional regulation field, such as the role of chromatin interactions. The main advantage of this technique is that it provides a relatively unbiased capture of the interactions for a locus of interest.
Although this method can provide insight into promoter-enhancer interactions, it can also be applied to identify other types of chromatin interactions.
Generally, individuals new to this method will struggle because it takes several days for the protocol, and the primer design requires trial and error and atypical primer orientation.
To preserve chromatin interactions in cells of choice, cross-link them by adding 9.5 milliliters of 1% electron microscopy-grade formaldehyde in PBS per 10 million cells. Incubate the cells while rocking for 10 minutes at room temperature. Transfer the reaction tubes to ice to quench the cross-linking reaction, and add ice-cold 1 molar glycine to a final concentration of 0.125 molar and mix by gentle inversion. After centrifuging and washing the cells according to the text protocol, resuspend the pellet in 125 microliters of 5 millimolar EDTA with 0.5% SDS and 1x protease inhibitors. Incubate this cell suspension on ice for 10 minutes. To make sure that the cell lysis is complete, mix 6 microliters of the cells with 6 microliters of trypan blue on a microscope slide, and cover with a cover slip. View under a microscope to see the interior of the lysed cells blue and unlysed cells white.